We'll continue explaining more techniques about the purification, detection, and characterization of proteins. And here we are going to start by talking about enzyme and antibody assays. Assays are laboratory tests that are done in chemistry, biochemistry, biology, mining, and the purpose of these tests are to identify or to quantify or to test the activity of a target entity. So this could be, in the case of chemistry, it could be a mineral, a molecule, a compound. In biology, it could be an enzyme, a protein, a DNA particle. So that's what we mean by an assay. In general, biological assays take advantage of the highly distinctive properties of a protein. These properties include its ability to bind a particular ligand, to catalyze a particular reaction, or to be recognized by a specific antibody. And these all three were all covered in the previous slide sets. So we know that proteins can bind to a particular ligand, and we can evaluate this binding through measuring its specificity and affinity. We also looked at enzymatic reaction and how they can catalyze a particular reaction. And we also mentioned how specific is the bond between an antibody and its antigens. Assays usually, when being designed, should be simple and fast because a lot of time it's the measurement of the concentration or the presence of certain biological entities is very time sensitive. For example, if somebody is sick and we want to find out what is causing the disease, how fast this person is being diagnosed could determine whether they survive or not. So for example, also another example, if somebody has a heart attack or we suspect that someone has a heart attack, we need to find that out quickly and not wait for days in order to administer the proper treatment. Assays also have to be sensitive and only require small amounts of proteins because in general most proteins that are detected for diagnosis or for biological phenomena understanding are not present in high quantities in the body. So we need the assay to be sensitive in order to be able to detect those small amounts of proteins that are usually present. The most common assays are chromogenic and luminescent enzyme reactions. The meaning of chromogenic is assays that result in a change in color. So for example, if an enzyme interacts with its substrate and the substrate is broken into a compound that changes, and that breaking results in a byproduct whose color is green, that means that the more substrate there is, the more green intensity we're going to get. So that's what we mean by chromogenic reactions. Luminescent reactions are those who result in the emission of energy in the form of light. And here we can measure how many light units there are, and of course the more light units there are, the more energy was emitted, so the more efficient the reaction is. Another form of reactions that we are going to discuss later is the ones that results in fluorescent signals, but this would be discussed in a couple of slides. So assays that involve enzyme reactions not only require the detection of whether an enzyme is present or not, but also its functionality. So how effective this enzyme and is it doing its job correctly? So this is what we mean by the functional aspect. Enzymatic activity assays detect the loss of a substrate or the formation of the product. So if we have an enzyme and we don't have its substrate, we're not going to end up getting its product. So we need to test the transformation from substrate to product. So substrate needs to decrease and product need to increase in order to ascertain that there is actually an active enzyme present in the reaction. Chromogenic substrates, as I just mentioned, are the ones that change color in the course of the reaction, and the change in color meaning a decrease in the substrate's color or an increase in the product's color. So it could be also measured either way. So only samples with the enzyme will change color. If the enzyme is not present, you're not going to get any change in color. So let's say we have A by an enzyme results with B, and A's color is red while B's color is blue. So if there is an enzyme, the amount of red or the amount of A is going to decrease and the amount of B is going to increase. If there is no enzyme, then the amount of red is going to stay the same and the amount of blue is going to stay the same. So if we detect the change in the intensity in the color red or the intensity in the color blue, we can check whether the enzyme is present. Antibody assays depend on the binding of an antibody to its antigen. This can be visualized by labeling the antibody with an enzyme or a radio-labeled or fluorescent molecule. So the way that works is that if we have an antigen and an antibody binds to it, we want to detect if this phenomena has occurred. Of course, using a naked eye and no labeling, we won't be able to see this happening. So what we do is tag the antibody. So if we have an antibody, we tag it here with a label. This label can be an enzyme, a radio label, or a fluorescent label. If it's an enzyme, then we can add its substrate and detect its presence if the reaction takes place or not. And usually the substrate is chromogenic, as we explained in the previous slide. If the tag is radio labeled, then we can use a scintillator and detect whether there is any radiation coming out of the signal or not. If the tag is fluorescent, we can detect the presence of fluorescence by subjecting the specimen with light at a certain frequency and detecting what light comes back at a specific different wavelength. So this brings us to the definition of fluorescence. So if a molecule is fluorescent and we subject it to a light at a certain wavelength, so let's say we subject it, it's going to absorb light and emit it back at a lower energy. It's going to vibrate that light back at a certain energy. So this is going to give me lower red value, because of course if it absorbs light at a certain wavelength, it's going to absorb some of the energy and emit the rest of the energy back at a lower energy. So if it's red, it's going to give us infrared, which is not a very practical example. However, if it's green, it could, for example, emit light back at a red wavelength, depending on how well it absorbed the energy. So we can detect whether this reaction takes place or not. If it does, that means that the antibody is bound to the antigen. If it doesn't, then that means that the antibody does not bind to the antigen, meaning that the antigen is not present if we're looking for a certain antigen. So the advantage of using an enzyme as a tag is basically it has a magnifying effect, because as I mentioned, you have one enzyme molecule, and if you add substrate to it, it's going to catalyze large amounts of that substrate, and because it has magnifying effect to its presence. However, if you have, so you have multiple substrates that have been catalyzed by the enzyme, as we mentioned previously. However, if you have a fluorescent molecule, then you're going to get signal from one molecule as opposed to many substrate molecules. So having enzymes linked to the antibody has an intrinsic magnification property to it, if you will. We can use also what we call a labeled secondary antibody, and the reason for this is usually purely economic. So instead of having the tag being, as we mentioned previously, being an enzyme or a fluorescent or radiolabeled molecule, what we can do is that we do not label the first antibody because there isn't enough of that antibody, but we add another antibody that is labeled, that what we call a secondary antibody. That antibody would bind to the primary antibody, and that antibody is tagged, and usually it's tagged with an enzyme. And the reason we do this is because, let's say we have an antigen that is not very common, so you need an antibody for it. However, because it's not that very common, the antibody is very expensive. So tagging the antibody is pricey, plus you do not want to use up most of that antibody. That's what we call the primary antibody. So the primary antibody that's specific to the antigen is going to bind to the antigen, but since it's not tagged, you won't be able to see it. In order to be able to see it, we have a secondary enzyme that could bind to any region of antibodies. So it can bind to a number of antibodies. And because of that, this enzyme is common, and this enzyme is a lot cheaper, and it's already labeled. So we use this secondary antibody, add it to the assay. If the primary antibody is present, it binds to it. And of course, it has an enzyme. We use the substrate and we get our signal. If the primary antibody is not bound to the antigen, then there's no place for the secondary antibody to bind, and we can figure out that there is. This is an example of antibody labeling. If we the labeling here is done using a GFP, which is green fluorescent protein, and this is actually a living specimen. So you can actually add the antibodies that are tagged with green fluorescent protein, then subjecting them to light, and the light would come back at a wavelength that would give you the color green. So you not only know the presence of the protein you are looking for, but you can also see its spatial distribution within the specimen. We can also detect proteins in gels. As we mentioned in the previous slide set, we can separate proteins in either 1D or 2D gel electrophoresis. So these proteins, when they are embedded in the gel, they are usually not visible. So in order to be able to see them, we actually label or stain the protein in the gel. We stain the protein in the gel, and these are the bands that you see in gel electrophoresis, the separate lines that we saw in the previous example. Or we can further analyze the proteins in order to recognize exactly what type of proteins they are by transferring the proteins from the gel to a membrane using electrophoresis. Again, we defined electrophoresis as moving the particles from one place to another by applying an electric field, which is exactly what we do here. As you remember, when we use 1D or 2D electrophoresis, the most information we can get is, if we use 1D electrophoresis, is the protein's mass, and if we use 2D electrophoresis, we can recognize its mass and its charge, but we cannot be 100% sure that a certain protein is present. In order to recognize what exact protein is present, we use a technique called Western blotting. Western blotting combines gel electrophoresis and the use of antibodies to recognize specific proteins that are present in our sample. So we separate proteins using 1D gel electrophoresis. Afterwards, we recognize the protein and identify what exact protein we have using antibodies. Usually in Western blotting, primary and secondary bodies are used, just as we explained a couple of slides ago. And we use a luminescent signal to identify what kind of interaction between the antibody and the protein has taken place. So these are the steps for Western blotting. At the beginning, we have an SDS polyacrylamide gel. This you can review in the 1D electrophoresis slide set, which is the previous slide set. So once the proteins are distributed based on their mass using SDS polyacrylamide gel electrophoresis, and again, these bands are visible only by staining the gel after the protein is distributed, or before. It depends on the technique used. So you have this gel, and we want to transfer the proteins from the gel to a membrane. So an electric field is applied from the gel to the membrane, and if you remember, all these proteins are treated with SDS, so all of them have a negative charge. So the gel has a negative pole while the membrane has a positive pole. This would cause the proteins to electrophoretically transfer from the gel to the membrane. So all the proteins are going to move in the same arrangement they are in the gel to the membrane, and as you can see here. However, proteins on the membrane are not visible because the staining is only possible in the gel. Once the proteins are on the membrane, we incubate the membrane with the primary antibody. So we put the membrane in a container that contains the primary antibody only for the protein we're looking for. If we see the gel here, there are multiple masses of proteins, meaning there is more than one protein present in our sample. In this case, we are looking for a specific protein. We use the antibody specific to that protein and incubate the membrane in for an hour or so. After that, we wash the membrane, so only the antibodies that are specific to the one protein remain on the membrane. And then we incubate the membrane again with a second antibody that is linked to an enzyme. That incubation also takes about an hour. Then again, the membrane is washed. After the washing, if we want to see whether the secondary antibody is bound to the primary antibody and that the primary antibody is bound to the protein, we apply either chromogenic detection or luminescent detection, depending on what techniques are available and what secondary enzymes are available. In this example, it's chromogenic detection. So if we add the substrate of the enzyme that is linked on the secondary antibody, there is going to be a change in color only at the place where the protein is present. And here you can see a line that has a different color from all the other lines, meaning that the protein that we are looking for is present at this location, and that is because the primary antibody is bound to it. The secondary body that has an enzyme linked is bound to the primary antibody, and that the enzyme has catalyzed the chromogenic substrate that we have used. Radioisotopes are also a possibility to label biological compounds, specifically proteins. In radioisotope, we measure what we call specific activity. which is the amount of radioactivity per unit of material measured in disintegrations per minute per millimole. So you know that radioisotope disintegrate over time, and as they do that, they emit energy in the form of radio waves. So we can measure the disintegration by measuring the energy that is emitted as the substance disintegrates. The specific activity is dependent on the probability of the isotope decaying, and if you remember from your previous classes and from high school, this is determined by its half-life, so how fast it's going to take a certain substance to for half of the amount present to disintegrate. The biological activity of labeled molecules with radioisotope may be altered. So for example, if we label an enzyme with a radioisotope, that enzyme's activity may differ because when we label it with a radioisotope, the energy emitted by that isotope could change its conformation, which would result in changing its activity. In order to measure the amount of energy that's emitted from a radioisotope, we have two options. Either a Geiger counter is used. A Geiger counter measures the ions that are emitted in the rays that are emitted from the isotope. These rays interact with a gas that is present in the Geiger counter, and that gas produces ions, and the Geiger counter counts those ions. Similarly, there's a scintillation counter. The scintillation counter also uses the rays emitted. However, instead of using a gas like a Geiger counter, the scintillation counter uses a liquid that contains fluorescent molecules. Once the energy is emitted from the radio label, they interact with this liquid, which causes the fluorescence molecule in it to flash lights when they absorb the energy. So the scintillation counter counts the number of photons that are emitted from the fluorescence molecules that are present in its liquid. A very important application of radio labeling are pulse chase experiment. And pulse chase experiment takes advantage of the fact that radio labeling provides a time component that other types of labeling do not. And what I mean time component is that, as we just discussed, the measurement of the radio signal emitted from a radio label is going to decrease over time. So we can see that at a certain period of time, the quantity of radio signal is very high, and that is going to decrease exponentially until the signal is very, very faint. So in the pulse chase experiment, what is done is that a certain amount of radio labeled amino acids are added to the cells, and when these radio labeled amino acids are added to the cell is going to use them to synthesize proteins. So you're going to have a bunch of radio labeled protein. The chase phase is when you do not add any radio labeled protein amino acids, but add regular amino acids that have no labeling. So you'll have a period of time where you have the cell produce a large amounts of radio labeled proteins. Then there are normal proteins in production. So here we want to see if a protein that's converted from a precursor to a mature protein happens in time. So there's a normal protein and a mutant protein. The normal protein, the protein is converted from the precursor to the mature protein. The mutant protein, there is no conversion from the precursor to the mature case. So if they add a pulse of radio labeled amino acids for half an hour, and then they add the chase, which is the unlabeled amino acids, for 24 hours, and they trace the presence of the radio signal over time. So for the normal protein, the beginning, the first half an hour, we have both the precursor and the mature proteins that are radio labeled. However, over time, there is no precursor that's radio labeled, only mature protein that's radio labeled. And if you see over time, the signal kind of gets fainter until 24 hours when the signal is very faint. What's interesting here is that only at the first half an hour we see radio labeled precursor, and all the radio labeled protein afterwards is all mature, meaning that all the precursor protein has been converted and that the radio labeled amino acid is only present in the mature protein. Here in the second experiment, when we have the mutant protein that has incorporated the radio labeled signal, we see that we have radio labeled signal that is decaying over time, but this is only present in the precursor. This means that the precursor is not being converted to the mature protein at all. And here, again, the advantage of radio labeling is that you can track the label over time, and this is not an option when using, for example, fluorescence labeling or enzymatic labeling or any other type of label. I'm going to add a video for the pulse chase experimentation, so if this is not clear, please watch it. Next, we're going to discuss how to decipher the primary structure of a protein. So basically, we want to understand what the sequence of amino acid that make up the protein is. So the first technique that has been used and is not very popular nowadays was called the Edman degradation. The Edman degradation depends on disconnecting the last amino acid of the amino acid sequence, because there's a free amino acid at the N terminal of the polypeptide sequence. So that last amino acid is labeled, then it is cleaved, so it's detached from the amino acid sequence, and it is identified using high pressure liquid chromatography, or which is called HPLC. HPLC basically can identify what sequence, what amino acid we have. It's a similar type of the chromatography techniques that we discussed earlier. However, it involves detaching that strong bond between the last amino acid and the sequence by applying high liquid pressure. After the detachment, that labeled amino acid is identified. Then the cycle is repeated again, because since the last amino acid is removed, the one before it would be a free amino acid. So that one is again labeled, then subjected to high pressure, then recognized, and so on. The cycle keeps repeating itself. However, as I just mentioned, this Edman degradation technique is not very common because it is very complex, and other techniques that we're going to discuss right now are much more simpler, and they are a lot faster. Nowadays, the most common technique for deciphering the primary protein structure is analyzing the DNA sequence that brings up the protein. So if we know the DNA sequence for each 3 nucleotides, we know the corresponding amino acid, so it's a lot easier to understand what the protein is and what its sequence is by analyzing its corresponding genome sequence than by analyzing it one amino acid at a time. Another technique that is also more popular than the Edman degradation, however it's also dependent on the protein itself and not its genome, is the peptide mass fingerprint. Here a bunch of digestion protein. We've mentioned proteases before. Proteases are enzymes that cleave polypeptide chains, so they break down proteins. So what is done is that the proteins that we have are mixed with these proteases, and these proteases are going to cleave and cut the polypeptide chain at different locations. Afterwards, that mixture of small peptide chains is analyzed for its molecular weight, and we know each proteolytic fragment we can look up in the genome database for similar sizes for different proteins, and from that fingerprint the different lengths of protein, which are usually analyzed using gel analysis techniques like how we mentioned earlier with electrophoresis. We get that fingerprint of different lengths of polypeptide chains, and from these lengths we look at the corresponding possible protein when using the specific protease mixture that we have used. X-ray crystallography is not used to decipher the polypeptide chain sequence. However, it's used to understand the 3D structure of the protein, so we know what the protein looks like, and as we have been repeating so many times, the structure of the protein dictates its function and would help us better understand how it accomplishes that function. So the most important part of X-ray crystallography is the ability to crystallize the protein, so to have it in a homogeneous construct that is stable, a 3D structure. So after the protein is crystallized, X-ray beams are passed through it. These X-ray beams are reflected off of the protein molecules, the molecules that make up the protein crystal, and from that reflection, which is dependent on the arrangement of the electrons around the atoms of the crystals, we can understand the diffraction pattern that results with discrete spots. So understanding these spots using mathematical equations that we will definitely not go through, we can decipher what the 3D structure of the protein is. The question here is why X-rays are used. The reason is that they have a small enough wavelength to be able to penetrate the protein crystal, and they would diffract off of the different electron densities around the atoms that make up the protein. The measured intensities of the spots that result from the X-ray scattering off of the protein crystals depend on, as I mentioned earlier, the distribution of the electrons, which is determined by the atomic structure of the protein 3D conformation. At the end of the X-ray crystallography, the result that we get is an electron density map that is further analyzed in order to get a 3D structure of the protein. So this is what we get. You have a protein crystal, an X-ray source. The X-ray beam hits the protein crystal, and it scatters off that crystal when it hits electron fields at different densities. So depending on the density of the electron, the refraction is going to happen. After the diffraction, the diffracted beams are collected on a detector. This detector could be a digital electron detector, or it could be a film, similar to the film that used to be used for photography. However, note that what we get at the end is an electron density map. These spots, the darker ones, are the ones that are a result of the X-ray diffracted hitting the film or detector. So if you notice, there are lighter spots and there are darker spots, which, as we just mentioned, depend on the electron density around the molecules. The other technique that is used to decipher the 3D structure of a protein is cryo-electron microscopy. Cryo-electron microscopy depends on having a 3D structure of the protein. However, instead of crystallization, as what takes place in X-ray crystallography, what we're depending on here is rapidly freezing the protein in liquid helium. This rapid freezing preserves the structure of the protein. It preserves the 3D structure of the protein. Then it allows us to examine this frozen sample in what we call a cryo-electron microscope. The final result from the cryo-electron microscope are pictures. These pictures are assembled when they are taken from different angles. They are recorded on a film. However, note that the cryo-electron microscopy is a type of electron microscopy. It's not a type of light microscopy because, as we mentioned earlier, light microscopy requires larger samples because the wavelength of light is a lot larger than the wavelength of electrons. So electrons are able to go through the small angles of the 3D structure of the protein, and then they are reflected to provide us with a 3D image of the protein structure. However, note that in cryo-electron microscopy, it's very important to use very low dosages of electrons. This is unlike scanning electron microscopy, which we mentioned for studying the structure of materials. We use very small doses of electrons because large doses of electrons would result in radiation damage to the protein structure and would provide false data. NMR spectroscopy is the third technique that we're going to mention to get the 3D structure of the protein. NMR stands for nuclear magnetic resonance spectroscopy, and here we get the atomic structure. So what are the atoms that make up the protein and how they are arranged in space. A concentrated protein solution is used for NMR spectroscopy. That solution is placed in a magnetic field similar to those used in magnetic resonance imaging. Then different radio frequencies are applied to those aligned particles in the magnetic field. This results in different nuclear spin states which are different because we have different atoms making up the protein. The spin states of the atoms are influenced by the distance between them and other neighboring atoms and their spin states. So we can tell if these effect of the spin states between two adjacent molecules is high. We can guess that these distances between them are small. However, the smaller the effect of one residue's spin state to the next, that means that the distance between them and the influence from one spin state to the other is a lot less because the distance is greater. We can guess from the magnitude of the spin effect, as I mentioned, the distance between the residues that can be calculated, and the final result is a 3D structure of the protein. The limitation of NMR spectroscopy is that it can only be applied to proteins with a size less than 20 kilodaltons, and we know that a lot of proteins have a much larger size than that. So NMR spectroscopy is used a lot to decipher the 3D structure, not of whole proteins, but of specific isolated protein domains that we have described previously in the class.